Hi and welcome to another BIOS E1A practice problems tutorial. This time we're going to be looking at number 19 from practice problems 2.5. This question is one about restriction enzyme usage for determining whether or not an individual has a mutant copy of a gene. The question reads, you are a doctor working with a young couple from a family in which a hereditary form of early onset Parkinson's disease is common. For this form of Parkinson's, having a single mutant copy of this gene is sufficient to cause the disease. The disease is caused by a missense mutation in the LRRK2 gene. This mutation interrupts a BAM-HI restriction site that is present in the normal sequence. Both the man and the woman have a parent with the disease, and they have come to you because they would like to use preimplantation genetic testing to have a baby that will not inherit the disease. The couple is able to produce 6 embryos via in vitro fertilization, and you use PCR and restriction digest analysis to do genetic testing on one cell from each embryo. The results are shown below. Which of the embryos would you recommend that the couple implant to ensure that their child is not affected by the disease? It then shows an image of a gel showing banding patterns for each of the 6 embryos. So what is really at the core of this question is parsing out which banding patterns we expect to see with a mutant individual compared to the normal individual that has a BAM-HI site in their LRRK2 gene. So for simplicity's sake, let's say that this is the part of the LRRK2 gene that we care about. It's obviously not the exact sequence, and in reality the gene is many more base pairs long. But we're only focusing on this one section for our purposes. We've already carried out PCR to amplify the DNA fragment we care about, and we can see here that the BAM-HI restriction site is outlined in red, and it's present in this copy of the gene. As a reminder, the restriction site is the DNA sequence that a restriction enzyme will recognize in order to make cuts in both strands of DNA. In this specific case, the restriction enzyme is going to cleave between the 2 guanines and the restriction site code on either strand, as is indicated here with arrows. The result is essentially a cut in the DNA as shown, and so the NO nuclease is going to come in and cleave the DNA at both of those locations, and the 2 fragments will no longer be joined. So here are the 2 resultant products. In this specific case, we say that each of the products has a 5' overhang because the 5' end of either strand extends past the 3' end. Generally, when overhangs like this are present, it's not uncommon for the 2 ends to reanneal and for DNA ligase to fix the break. But for the purposes of this class and this question, we won't get too into the weeds on that, and we'll just observe that the restriction enzyme results in 2 shorter products, as shown here. So what happens if we run these 2 products on a gel? Because there are 2 products of different molecular weight, we're going to get 2 distinct bands. The shorter product is going to travel farther along the gel as it encounters less resistance from the pores of the gel. The larger restriction enzyme product doesn't travel quite as far, as shown here. So in the context of the question, this is what the banding pattern would look like for a non-mutant individual. This is what the couple's looking for. Both copies of the gene, one from either parent, would have an intact restriction site, and the banding pattern would look like this, with 2 distinct bands, each corresponding to one of the 2 restriction products. So what would it look like if there were a mutant that did indeed have a mutation in the BAM-HI site? Let's arbitrarily say that there is a mutation in the first base pair of the site, and thymine is substituted for guanine, as shown here. This means that the normal restriction site cannot be recognized by the restriction enzyme and no cleavage happens. The gene stays fully intact. So in an individual with 2 mutated LRRK2 genes, we're going to see no smaller restriction enzyme products. They'll all be the full length. So on our gel, let's consider what an individual with 2 mutated LRRK2 genes will look like. Again, if both genes are mutated, no restriction products are produced, and all the DNA fragments obtained from PCR will remain intact and be larger than the fragments that we found in an individual with 2 normal copies of the gene. Because these non-cleaved products have a greater weight, they won't travel as far down the gel, and the banding pattern for an individual with 2 mutations will be shown as it is on this slide. Each gene is the same, so only one band will actually appear. Now let's think about the banding pattern for an individual with one mutated gene and one normal gene. The normal DNA will be cleaved by the restriction enzyme, and the bands for the products of the enzyme digest will look just the same as in a normal individual. You'll see those same two bands. The mutated gene is also going to be present, though, and this DNA sequence will not be cleaved by the restriction enzyme, producing the same band as we see in the double mutant. So we can see that an individual with one mutant gene and one normal gene has a banding pattern that combines both banding patterns that we found before. So now that we know what each possible genotype looks like on the gel, let's compare those to what we saw in the question to see which individual would not be infected with the disease. So we want an individual that's just normal and has those two bands that we get from the restriction enzyme digest. Comparing the gels, we can see that none of the embryos tested has a completely normal banding pattern. That is, there is no embryo that has two and only two bands that correspond to the genes. Embryos 2 and 4 have only one band, indicating two mutated copies, as we laid out earlier, and embryos 1, 3, 5, and 6 all have three bands that indicate that they have a single mutated copy and a single normal copy. Because in this instance, even a single mutated copy is enough to cause the disease, none of these embryos is going to be free of the disease. I know this isn't really the most uplifting outcome to this problem, but hopefully this video is able to help in your understanding of it. Best of luck while you continue your studies, and good luck on the exam.